Hang on, I just went too fast. Ah, how do I go back? Okay, well, it's not letting me go back, so I'll just start here. So I'm going to talk today about some work that a group of us are doing on wire-tailed manakins. This is a group of birds that have been well studied by Brant Ryder for the past 15 years or so in the Tipitini Biological Station in Ecuador. And they are very interesting because they have exploded leks. They have no paternal care, as you'd expect, but most interestingly, they form cooperative coalition partnerships. And so when Brant and I started working together on this project, along with Brent Horton, we got particularly interested in the role of hormones in their behavior, specifically in their cooperative behavior. Brant had done work for his previous work, is starting in grad school, on their networks and their social networks. And these are the basis, these cooperative partnerships are the basis of the social networks in the species. They have a social hierarchy, so they start off as formative floaters, and then over a period of years, their plumage changes to a definitive plumage as they start off green like the females, and they gradually transition into the more spectacular plumage. And sometime in their second to third year or so, they can acquire a territory. The coalition partnerships occur when these males display on their territories and another individual male will cooperate and participate in this display. So in this case, we have a territorial male here in the definitive plumage, and then next to him is a pre-definitive floater who's visiting the territory. And these coalition partnerships are consistent and occur across years. And this is, again, the basis of these social networks that Brant had studied for many years. So this is an example of a lek right here. And the gray individuals are territory holders. The green individuals are females that visited the lek. And the red and yellow individuals are floaters. And you can see that through his observations, he was able to develop an idea or a pattern of who interacts with whom, how often they interact, and such. And so when Brent Horton and I started working with Brant on this project, our idea was to try to look at the mechanisms behind these social behaviors. And basically the idea is on the left here, we have an example of testosterone. So we're looking at hormone-behavioral or hormone regulatory networks, and then how they scale up to influence social behavior, which I'll talk about how we measured social behavior in a minute. And finally, what I'm not going to talk about in this talk is how this eventually scales up to social network structure. So let's first talk about the hormone regulatory network. And again, specifically here, I'm talking about testosterone. Tomorrow, Brent's going to be talking more about the hormone regulatory network, specifically looking in the brain. So our first study was done almost a decade ago now. And what we did is we looked at testosterone levels across these different age classes or different classes. So the gray bar is a definitive territorial. And what we found is that they had higher testosterone levels than any of the different floater classes. But what's most interesting to me was, well, first of all, it's interesting to see this difference between the classes. But most interesting was the individual variation. And that's something that behavioral endocrinologists have traditionally tried to ignore. But any evolutionary biologist would tell you that individual variation is obviously very, very interesting, and the basis of which selection can act upon. And so here you can see there's 5 individuals here that have testosterone levels that are much higher than the rest of the territorial individuals. Likewise, amongst the floaters, these next 3 bars, you can see there's a couple of individuals here that have testosterone levels that are much higher than the others and are actually in the realm of what we'd see normally for definitive territorial individuals. We took this a step further, starting a few years ago, specifically with Ben Vernasco and Roz Dakin helping us. And what we did is we sampled individuals multiple times per year and then across multiple breeding seasons. And the idea here was to try to develop a better idea of their hormone, the consistency in their hormone levels, to see if we could see if they have actually a hormonal phenotype. And so what we found here, now this graph's a little bit confusing. On the y-axis, you have testosterone levels. And then on the x-axis, we have individuals sorted by their mean testosterone score. So like I said, some individuals were sampled as many as 8 times. And so we sorted them by their mean score. And what you can see here, first of all, is that there's a lot of variation in testosterone levels. You can also notice that the pink bars, which are the floaters, are arranged at the left side of the graph, and then the gray bars tend to be on the right side. So again, the territory holders, the gray bars, have on average higher testosterone levels than the floaters. But again, you see a lot of individual variation, right? Especially through the middle section, where you have some territory holders with lower testosterone levels and some floaters with higher levels. The other thing I want to point out here is that we found that testosterone levels were repeatable. It was about 19% of the standing variation was explainable in the analysis. So we do see a hormonal phenotype. Okay, how does this relate to the behavior? Well, first, to investigate the behavior, the social interactions between males, we use proximity data logging. So that involves capturing males and putting a coded nanotag on their back, and then subsequently putting data loggers on the territories. And then we set the gain on the logger such that the radius encompasses the whole territory. And so that way we would collect a lot of data on who visits whom and how much time they spend on their territories and how much time they spend on other individuals' territories. And so again, we could collect a lot of information that way. This is, we tagged about 120 males per year. And then again, we would set up these, the receivers on their territories multiple times per breeding season. And as you can see here, collect a lot of information that way. And from that, we could develop and build these social networks. This is an example of 2 LECs, 2 social networks, like A and like B. This doesn't show up, but there can be weighted by the frequency of the male interactions. And we can see who interacts with whom. In this case, we also have these 3 yellow individuals are actually floaters that link the two LECs together. We measured 3 behaviors to define a male's cooperative behavior. First, we measured the effort. This is the number of pings on the displayed territories per day. So in other words, how much time are these individuals spending on their territory? You can see here in this graph that, unsurprisingly, territorial individuals tend to spend more time on the territories than the floaters. We also had strength, measured strength. This is the number of social interactions that a male had per day. And finally, degree, the number of partnerships a male had per day. So the number of different individuals he interacted with. Okay, so now, since we have the idea of what the hormones look like and the behavior, we can try to link the 2. So in floater males, testosterone was positively associated with effort and strength. So in this top graph, you have effort. So this is the number of pings per day. And you can see the y-axis, the x-axis is testosterone levels. And you can see a positive relationship. That is, males with higher testosterone levels spent more time, spent more effort than males with lower testosterone levels. We see the same positive relationship here with the strength, with number of interactions. So higher testosterone are having more interactions per day. With territorial individuals, we actually see the opposite. So here we see the top graph, efforts, actually a curvilinear relationship. but individuals with high testosterone were actually spending less time on the territories. Interesting enough, here they also had fewer interactions per day. So again, a negative relationship. So individuals with high testosterone had fewer interactions per day. So what you can see then is that this hormone behavior relationship is status dependent, right? So we see positive relationships between testosterone and the behaviors in floaters, and negative relationships in the territory holders. We actually followed this up by doing an experiment where we took territory holders and implanted them with testosterone to make the testosterone levels higher. And you can see here quickly, the blue bars, the left side of the blue ones or points are testosterone levels, or sorry, behavior before implant, and the right hand are testosterone levels after implant. And you can see in strength and degree that when we elevated their testosterone levels, we saw a negative effect. So it decreased the number of interactions per day, and it decreased the number of partnerships. So going back to our question, can circulating testosterone levels explain status and individual variation in social behavior? Yes. In floater males, high testosterone promoted social behaviors necessary to gain status and eventually a territory. But in territorial males, high testosterone negatively impacted those same behaviors, same social behaviors. And we see that both observationally and experimentally. So with that, obviously I'd like to acknowledge my co-authors that actually did all the work, specifically, or particularly Roz Dakin, as well as the collaboration I've had with both Brent and Brant through the years. The field crews that did all the real work, and then our funding sources. And with that, I'd be happy to take any questions.